Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today, we'll be talking to Dr. David C. Weindorf. Compost needs to be regularly tested to ensure labels regarding organic matter, nutrients, and more remain accurate. However, getting compost tested is time and cost intensive. This episode, David joins me to discuss how developing a model to predict organic matter from cell phone pictures might make this process a whole lot easier. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Dr. David C. Weindorf with us. Dr. Weindorf currently serves as the Vice President for Research and Economic Development at Georgia Southern University. He holds a bachelor's in range management, a master's in soil science with a geochemistry minor, and a PhD in agronomy from Texas Tech University. He's a fellow and presidential award winner of the Soil Science Society of America. As a scientist and Fulbright scholar, he has worked in over 30 countries and published more than 220 peer-reviewed research papers in top international journals germane to soil, water, and compost science and environmental quality assessment. Hello, David. Welcome to the show. How are you doing today?
I'm doing great. Thank you so much for having me. I really appreciate it.
Yeah, we are thrilled to have you today. So today we're talking about using pictures to test compost quality. So to get us started, can you just tell us a little bit more about the problem your research was hoping to address, kind of what makes a good compost, how we can tell if it's good compost, and kind of some background that led up to this research?
Absolutely. That's a great question. So, compost is a soil amendment that farmers and homeowners have used for years to benefit soil quality. One of the things about compost that is different from chemical fertilizer is it is inherently variable. That means that bag number 1 and bag number 500 may be slightly different from each other. So when you buy a bag of commercial fertilizer, you have those 3 numbers on there, right? That indicate the N, P, and K value. But you don't have that on compost. So in order to get around that, what producers do with compost is they regularly test that. And that testing is performed in what's called a Seal of Testing Assurance Laboratory. That's an entire program that is conducted by the U.S. Composting Council. And it basically provides some physical and chemical properties of the compost and lets people know, what is the organic matter content like, the pH, the salinity, those kinds of things. So while that program has been very effective, because the compost is variable and in fact changing, in the bag over time, as it's heating and cooling and drying and wetting and those kinds of things, these results from the Seal of Testing Assurance Lab are only valid for a period of time. And then the pile has to be retested again, right, to see if it's changed at all. So when we do this type of lab analysis, it's expensive and it's time consuming. And so what we're trying to do is move more towards sensor-based approaches where we can collect very good quality data to characterize the compost, but do it more quickly, more efficiently, and less expensively than with traditional testing.
Sure. So I think for me, as just an average Jane walking around on this earth, my understanding of compost is mostly like throw your food scraps into a pile, and it would never occur to me to test that as just a person. So follow-up question is who is getting this tested? Are these compost companies? Are farmers doing this to make sure that, they factor that in properly with their fertilization rates? What is the scale of this testing going on?
That's a great question. And so the answer to that is both. Producers are doing this type of testing as well as the people receiving that type of compost, especially in large commercial applications. So think about it this way. A compost producer might be producing this compost from something like manure or biosolids or lawn and landscape debris or food scraps, things of that nature. And they put it in these long windrows and the pile heats up and aerates and gets turned over time and is degrading. So they want to test that and monitor that regularly throughout that entire process to make sure that you arrive at what we call finished compost, that kind of dark, crumbly, earthy kind of material that we really like to mix into our gardens. But in terms of the people who are purchasing this compost, imagine a commercial application for something like a a roadway right of way, right along a roadside when it's being constructed. They might put a compost blanket there, and they might be bringing this compost in by the 18-wheeler full, right? I mean, these large semi-trailer truck fulls, right? So when they get that on site, a lot of times an inspector from the Department of Transportation will want to verify that they are in fact receiving what the seller has said that they will be sending, right? And so having these types of sensor approaches provides a quick way that they can make that assessment on site as well.
Yeah, I guess if you were sending a trailer full across the country, that would certainly, being under a lot of heat or cold, depending on the time of year, would have a big impact. Sorry, my brain is very tempted to just completely derail this to talk about that. But we're not going to do that. We're going to show self-control. So, moving forward with this research, which I am very excited to talk about. So, you were hoping to kind of, as you mentioned, make this much easier for people to test, and specifically do that with kind of analyzing photos that people could take. So we're going to talk about the methods, but before we dive into that, I just want to define a very super cool sounding test that you can do, the loss on ignition kind of factor. So can you explain how that works?
Absolutely. So in terms of the research that we're talking about today, we're really focused on one specific parameter of the compost, and that's the organic matter content, right? And organic matter in compost works a lot like organic matter in soils. It is the organic constituent. So with loss on ignition testing, it sounds fancy, right? But it's really a very simple test. You dry a sample, to get it oven dry to make sure there's no moisture there. Get a weight of that sample. Typically you put about 10 grams in a crucible, and then you burn it in a furnace at a very high temperature, typically around 400 to 500 degrees for a few hours. Then when you take that out, you cool it off and you weigh it again. And what you find is all of the organic matter that was in that sample has been turned to ash. And because it's turned to ash, it now weighs, less than it did initially. And that difference in weight from the initial weight to the ashed weight is what allows us to define the loss on ignition organic matter content of the sample, both for soils as well as compost.
Okay. So that's kind of it's kind of your control measurement of the organic matter, which you could compare to the results from your photos.
Exactly.
So, and also, can you just define like what all is included in organic matter? I mean, obviously not like minerals, but like what would be included in that?
Yeah, and organics can take many different forms, and they derive from their feedstocks. That is the original source material from which they are broken down. So again, these feedstocks might be things like food and vegetable scraps. It could be fish scraps along the coastal areas. It could be manures of different types. It could be municipal biosolids. It could be lawn and landscape debris, mushroom waste, chicken litter, all kinds of different things. And those can all serve as feedstocks, which are then composted into the ultimate material, which is sold to the consumers.
Perfect. Okay. That ties in perfectly then with my next question is just how you went about this research. I know you collected a lot of samples, and I am keen to know just all about how that process went.
Yeah, absolutely. So we actually collected samples both in Canada as well as in the United States. And in Canada, in Ontario, we just went to a local Home Depot and we collected some bagged compost samples there, just like you would purchase here in the United States. But we found that the compost you purchase in Canada is really different than what you have in the United States. So there was compost derived from mushrooms. There's compost derived from chicken, sheep, and cow manure, different, proportions. And so really interesting stuff. And so we were able to collect some of those samples. And then in the United States, we went and got some of those bagged samples from multiple different states. This would include Georgia, Illinois, Texas, and Michigan. We also went out and collected some actual piles from windrows of commercially produced compost and collected those samples as well. So it was a really robust sample set collected from a wide geographic area, both in the United States as well as Canada.
I love this because, first of all, longtime listeners will know I am a huge fan of just like bootstrappy kind of science, because I think sometimes people are like, oh, they're just like in a lab coat and they get everything like shipped to them. And honestly, I just assumed you had people ship you stuff. So I love the idea that you're like, no, we just went to Home Depot. Like, of course, that's what we did. I love
Yeah,
that.
and we did. I mean, we did have some friends ship us some samples, but I made that sort of strange phone call. Hey, can you help me out here? I need you to go buy a bag of compost at your local Ace Hardware, whatever. Take a picture of what it is, like on the bag. I need to know exactly what it was, and then send me like a Ziploc bag full. That's all I need you to do. And so that's what we did. And so we went out and collected some of it ourselves, but we also had some friends help us as well. For that reason, I will tell you also, my wife and my daughter are in the acknowledgments of this paper because they were involved in the process as well.
Oh my gosh, that's so awesome. I love that.
That's right.
This research is so delightful. Yeah, so man, and wow, what a travel if you're in Georgia to get all the way up in Canada. I love that. So I believe you said you had like 150 samples. Am I making that number up, or is that correct?
It was 157 for this study.
Wow. Yeah. Okay. So you had all these samples, and then you went out and were doing your research on them. So can you tell us more about how that process worked?
Yeah, absolutely. So the first thing we did was take a small portion of each one of those samples, set it aside, and do the traditional testing through the loss on ignition testing. We did that in a normal laboratory with normal laboratory equipment, just per the normal guidance of the TMECC protocols. But then for the imaging aspect of the paper, which is the novel research that we're working on, we created a custom imaging box. And what we basically did was take a little plastic tub and we spray painted it completely black so there would be no outside light coming in. And then we took just a standard Google Pixel smartphone, cut a little hole in the top of this bucket, set it down on top of that. We would put compost in there while it was dry and put the cover on it, have the camera take a photo of it with the flash on, and then we would remove that cover, spray a little bit of moisture onto it, put the cover back on, and take a second photo. So we had photos of the compost dry as well as moist. And so then once we had all of those images collected and we had the loss on ignition data, we set about to start to do the statistical analysis and create the models that would then allow us to predict organic matter content directly from the images themselves.
Yeah, there is so much to dig into there. So I guess my first question is, so you had your little container was like black on the inside. So did you guys have to like test different like iterations to figure out which worked best? Like did you initially just, throw it in a cool container and then you're like, oh, the light is weird or the color is weird? Like what got you to that setup?
Yep. So that's a great question. One of the things that we had to be concerned with was the height of the container above the sample itself. Because just like if you take a smartphone and you hold it too close to something, it gets blurry, right? Mm-hmm. So we wanted to have it high enough up off the sample that we could get a decent image of it to where it was not blurry. So that was one of the considerations that we took into account.
Oh, I love that. Yeah, I didn't even think about that. Oh, that's so cool. I love this. So after you took your photos, then you had your model built out to try and determine the organic matter based off of just these pictures and this model you developed. So what did you find? How did it work?
Yeah, so I hand all that data off to the statisticians on our team. And these are people who are way smarter than me. And so I can't get too far into the weeds on the models, but what I can generally tell you is it works sort of like this, right? When you take a smartphone image, everybody's familiar with RGB, right? Red, green, and blue is the colors, right? Like you'd see on a television set or something like that, right? So all of the pixels that describe the image that you have taken are defined by those RGB, red, green, and blue colors. And we then converted those into something called hue, saturation, and value, HSV numbers, right? And for our soil scientists out there or others who are listening who are familiar with a Munsell color chart, it works sort of like that, hue, value, chroma, right? Then in addition to those data, and all of that data is numerical, let's remember that, it's all numerical data. In addition to that, we converted the geographic location of where the samples were collected into numerical format, and we used something called random forest modeling in order to put all of this data together. Now, one thing it's important for your listeners to know is If you know anything about statistics, I've always been told with enough data, you can prove anything. Okay. I mean, really. So what we call that is overfitting the data. When you have too many parameters in there, I mean, if you have 1,000 parameters, that's a model that you can prove anything with, and it's way too complicated. The trick in statistical modeling is to find the fewest number of variables that define the parameter of interest. That's the sweet spot. That's what we're looking for, right? And so that's what we did in this paper. Was we kind of looked at all these different variables, and then we identified which are the ones that are most important in describing the organic matter content of the compost. And as it turns out, two of these factors that we considered were really key among these models, right? One of those was the standard deviation of hue, one of the components of color, and the other was geographic location. So in fact, whether or not that sample came from Canada or West Texas from cotton gin trash made a big difference in terms of us being able to predict the organic matter content in the model. And what our results showed ultimately was we were able to more completely predict organic matter content in the dry samples as compared to the moist samples. And there's some reasons why that is the case.
I love that. For one thing, it feels like that definition of statistical data and how you approach it correctly encapsulates something that has always fascinated me about science in general, which is like every time we have a research thing on here, I'm like, well, like, what about this? Or like, why, like, how do you know that this isn't affecting this other thing? And so it's always fascinating to me how researchers kind of have to narrow down, like, we think these are the two things that are connected to each other that we're testing, or, you know, we can't test like 18 different things at once sometimes because it's too complicated. So this is great to finally have that codified in language for myself. So
yeah, I mean, there's a classic question that is asked of every doctoral student out there, and it goes like this: do you believe that correlation equals causation? Because as statisticians, we're always looking at R squares, and the higher the R square, it must be important, you know, right? What you can find is that the color of shoes worn in Fiji is related to the number of thunderstorms in New York City, right?
Right.
Those may be correlated, but that doesn't mean one causes the other. And so that's what we have to be careful of in statistics.
Yeah, I love that. And then this brings me to a question that I had about compost in general when you were speaking, so my understanding, and please soil science people, don't come at me if I'm wrong, I'm doing my best, is that soil is like when it's like chilling in its natural environment, and then once you like move it, it becomes dirt. So I always tell my uncle, who works at Home Depot, coincidentally, I always joke with him that like potting soil like doesn't exist. It's potting dirt. So I was curious because I feel like compost is like throw a bunch of scraps into a thing until they turn into dirt or soil. So I was curious about how different soil types, which would naturally have more or less organic matter, would factor into this. And it sounds like, because I was like, it's not like you're testing just a regular soil type. So I'm curious how this whole location-based results thing, like, why? Like, do we know why that matters? Where it's
Sure.
from?
Yeah. No, those are great questions. You know, to come back to your other point there about soil versus dirt, the way that I've heard it described is dirt is soil out of place. Right.
Okay.
So when it's in its natural environment, it's soil. But when you get it on your shoes and track it in your house, that's when it becomes dirt. It is soil out of place, right? Now, in terms of these organics being produced out of the soil, so to speak, in a commercial sense, and then being reapplied, the way that I like to think about it as a soil scientist is Mother Nature had this figured out 10 million years ago, right? Like leaves fall off the tree, they get moist, there's some bugs walking around in there, you know, chewing on stuff. It starts to rot down, and it forms this layer of high organic matter right at or near the Earth's surface, right? Those kinds of natural systems have been going on for millions of years, right? What we're talking about with compost is basically doing that same process, but outside the natural system of the soil. So we're putting on a concrete pad. We're controlling how much moisture goes into those organics. We are turning the pile regularly in order to aerate it to make sure that it doesn't go anaerobic and cause problems there. So we're sort of managing that decomposition process, sort of maybe taking the controls from Mother Nature a little bit to accelerate the process. And then once it reaches that sort of steady state at the end, where it's no longer undergoing active decomposition, that's when we bag it up, sell it, put it on the 18-wheeler, put it out in the final location. But with regard to soils that have a lot of organic matter in them, of course, one of the things that your listeners will know about are histosols. And these are highly organic soils, right, that have formed naturally, over, again, millions of years. And these histosols are one of the 12 naturally occurring soil orders as defined by U.S. soil taxonomy. But another mineral soil that has lots of organic matter in it are our mollisols. And those mollisols are what we often see across the grasslands, throughout the central United States. And those soils have high natural fertility in large part due to the high degree of organic matter that are in the upper mineral layer of those soils.
Okay, so then if we're in, and I will not even dare to attempt to place these geographically, but if we had like a really clayey soil versus like a really sandy soil, mm-hmm. is that going to affect then, it seems like that is then affecting the compost, and that is how you can kind of know?
It certainly could, yes. When the compost is mixed into the soil, the physical properties of the soil will interact differently with that compost. Specifically, you mentioned clayey soils. Clay particles, by and large part, many of them are flat. They're little pancake-like layers of soil, kaolinites and montmorillonites and things like this. So what happens is the organic matter tends to work its way in and get protected and sort of coated by that clay, and it protects it from degradation, from further degradation. But sands, for instance, larger particle size, there's really no affinity to that organic matter, and it can break down pretty quickly in those sandy soils. So yes, there is sort of an interplay there between soil physical properties and compost and how quickly it'll be broken down within the soil.
So you mentioned that the moist samples didn't do as well as the dry samples. I want to just loop back to that. Can you tell us more about why that is?
Yeah, that's absolutely the case. And we really feel like within the modeling, there were two reasons for that. First of all, when we applied the moisture to the samples, it was not the same amount of moisture in every sample. It was just somewhat subjective. And we did that purposefully because we wanted to reflect real world variability in moisture content. When you're, taking a scan or an image of a compost pile in this location or that location, the moisture is not going to be the same. So we just sort of haphazardly put moisture on there a little bit, here, more there. And so that caused some discrepancy in the model. The other issue was when we applied that moisture and then used the camera flash to image that compost sample, there was some reflectance coming back from the moisture, almost like a mirror. And it would reflect some of that as like a bright, reflection off of the moisture. And I think that also compromised some of the model performance for the moist samples. So for this reason, using the dry samples is definitely the way to go. They're more accurate.
Hi, everyone. I hope you're enjoying the show. Interested in learning more? David's paper, Towards Sustainable Compost Use, Prediction of Organic Matter via Smartphone Image Analysis, published in Agronomy Journal, will be freely available for the next 2 weeks. If you are a certified crop advisor or certified professional soil scientist, you can take a quiz for continuing education units for this episode, which can be found in our show notes or on certifiedcropadvisor.org. Let's get back to the show. Okay, cool. So you figured out that you could tell pretty consistently, in more so in the dry, and depending on the region, you could kind of pretty well I don't want to say guess, but I get like calculate.
Quantify.
Quantify, yeah. The levels of organic matter in here. We've talked about how soil types can affect it, and some of the patterns you found. So in your worksheet I had you fill out, you said that was about 75% accuracy,
That's correct.
which is really awesome and impressive since
Hmm.
it's a picture. It's
Yeah.
just crazy to me. So I would like to clarify what is meant with like 75% accuracy, because in my brain I'm like, oh, like 3 out of 4 times it's like spot on, and I have to be like, no, it's probably like we're 75% sure it's around here is probably more accurate. So can you talk about what is meant by 75% accuracy?
Yeah, that's the R squared that we come up with in terms of our validation testing. And I'll come back to that in just a second. But to your point, that sort of accuracy, like how spot on are we with those results, that's really defined in terms of root mean square error. And I don't want to get too far down in the weeds with you on all the statistical stuff, but the idea is if this is the point, there'll be a little variation above that or below that, right? And so it's sort of like a plus or minus, if you want to think about it that way. This is the value plus or minus a little bit, of give on either side. But one of the things I want to mention, you're talking about the accuracy here, is in statistical modeling, what we often do is take the full data set, that entire 157 samples that we were talking about, and we divide that on the front end into a calibration data set and a validation data set. So we take 70% of the samples at random, just randomly grab 70%, and we use those to build the model, right? Then once we have the model built, we take that and apply it to the other 30% and see how accurate the model performed. Generally, what you find in statistical modeling is the validation data set performs less robustly than the calibration data set, and that's important. When we publish these results, we always want to publish the validation results. They're the more conservative estimate, right? The actual model itself probably performed better than 75%, right? But the validation of this is really more conservative, and it gives us a really good understanding of how will this really perform in the real world.
Sure. And I also want to just ask about, like, because I think, a model, I would assume, is never going to be like 100%. Like look at like weather. Weather is never like, oh yes, it was exactly this.
Mm-hmm.
Even the day of, it's just too complicated to do that. So, like, when you say 75%, I'm imagining, based on some of where this is going, that's like pretty good, right? Like, what is like a successful model? Is there an average number for that, or?
Great question. And it really depends on context, right? If you were doing open heart surgery and you said 75% of the time it works out and 25% of the time the person dies, that wouldn't be very good for that application, would it?
Right.
We want a 99.9% success rate, right? But I make a little light of this, but the truth of the matter is, in soil science and compost science, I don't have to have 100% accuracy all the time. That's not really that important. I need to know, does that have a lot of organic matter in it or not so much, right? I mean, does it matter if it's 5% versus 5.5%? In my applications, no, it really doesn't. I mean, there are occasionally a few thresholds that we have to sort of come up against, right? And for those really important thresholds, we need to take the sample back to the lab and quantify it, in our traditional methods. But for an inspector, for a compost producer to make a quick check in the field and say, I wonder, is this compost that we're getting any good? Let me just take a quick snap or two of these photos and make a quick assessment. Oh yeah, it looks like it's, 27% organic matter. That's really good, I think those kinds of things and many of these types of agronomic applications are what people are looking for. Just sort of a yes or no, good versus bad, pretty good. They need sort of a qualitative assessment that's backed up by some sort of a semi-quantitative assessment as well. And that's what we're providing here.
Yeah, I love that. Tying that back to the goal application, I think is really helpful, because it's trying to replace the need for, this really expensive regular testing to just give people a ballpark, looking out at your garden and be like, things are wilty, we need to add water, like
Right.
it seems more like it's that level. I mean, obviously significantly more complicated and harder to tell just from your eyeballs, but
Yeah.
it seems kind of like that vibe, which is
So
great.
imagine this. Let me give you one other example of this. Let's say there was a certain amount of organic matter that you had to have in the compost in order for it to meet the criteria for your needs, right? If we could take just a quick photograph of that and certify that the level was above that or it didn't meet that, that's all we really need, one way or the other, right? And so if we're really concerned about exactly how close it is to that threshold, we can always go to the lab and run a traditional analysis. But that quick sort of yes or no decision, backed up by some of these proximal sensing and image analysis technologies, I think there's a real utility for those in the applications world.
Yeah, totally. Yeah. Oh man, I'm being such a fangirl about this research right now. I also I don't math, I don't statistics, so this is just like diving into like a fantastical new Willy Wonka world for me of wonder. So to contain that a little bit more, future research, you are moving forward with this to try and kind of get it more to the people. So tell me more about what are the next steps for this research and or other things related.
Yes, I think there's a couple of different ways that we can continue to enhance this model, maybe make it even more accurate than it is now. And I think that's really the ultimate goal, is to try to, continuously improve it, so to speak, right? One of those things would be to develop regional models. So you'll notice in the research that we did, I mean, again, we started in West Texas, we have some cotton gin trash there, all the way to sheep manure in Canada, right? Like lots of different kinds of things, right? And the organic matter content across all those samples ranged from around 14% to almost 80%. So big, wide range, right? If we were to create some regional models, right? What if we wanted to do a specific model for bagged compost sold in Canada, or a specific model for use in arid regions of West Texas, New Mexico, and Arizona, right? I think we could have some, more accuracy that are more accurate models, so to speak. I guess would be a good way to say it. In addition to that, also for this research, we used a particle size of 2 millimeters. We sieved all of the compost past 2 millimeters. And what we found is sometimes when we took that image, there was a little bit of a shadow from those particles that was cast on the soil or the compost below it. So if we used a finer screen size, maybe 1 millimeter, we might eliminate some of that shading and also see some improved accuracy in the models. But the ultimate goal, to get to the final part of your question, is to develop a cell phone app for this, which I think would be so cool. And to any of your listeners out there, if you're an app developer and you want to come help us out with this, I don't know the first thing about apps, they're just fun to use. But I think there's a really cool way that we could do this, and it could be a win-win, right? We develop a very simple app, get it out there to compost producers, to homeowners, whatever. You can just download it from the App Store. You take a picture, put in your credit card number, and for 99 cents or something like that, it sends that photo off, plugs it into our algorithm. We predict the compost organic matter content, deliver the results in 20 seconds, right? And you have the answer right there on screen. That would be such a fantastic, service to offer to people, right? Just a quick, inexpensive way to get that result out there. And would be a really nice way to share that with the general public.
Yeah, absolutely. That's wonderful. Please reach out if you have that skill set or know people that do. Which I think is a good transition then into our final 3 questions. So the first one is, if listeners would like to learn more about any of the things that we've discussed today, where can they go?
So I think one of the great sources of information will be the Compost Research and Education Foundation, and you can find their website at compostfoundation.org. And in addition to that, there's a number of journals that publish this type of research. I'd encourage your listeners to go read some of those journals. Compost Science and Utilization is a really good journal for this type of information. There's another one also from Elsevier called Waste Management, and you might look for that. And then again, the research that we just published, this particular paper is in Agronomy Journal, and they have some great compost content as well. And we did that purposefully to try to, get it out to a different audience. We really wanted to see if farmers could benefit from some of this type of stuff. So, reaching out to different audiences in different ways, I think is a really healthy thing to do.
Yeah, absolutely. Thank you for doing that and choosing us for your work. So then the next question is, if listeners would like to get involved, aside from app developers, what can they do?
So I think part of this is when they are using compost products, be informed consumers, right? Ask for that seal of testing assurance report to know exactly what's in that compost. You want to make sure that it doesn't have heavy metals in it. You want to make sure it's low salinity, that it has a high organic matter content, that the pH is okay. Know the source material of where it came from, right? Those are important questions to ask, whether you're at the garden center or going to a large commercial producer as well. They should have those types of reports available, and that will help the consumer decide or the purchaser decide if that compost meets their specific needs. Because not all composts are alike. Some have wood chips mixed into them to prevent erosion. Some are just, fine compost that is more nutritive in its concentrations, things of that nature. So,
yeah, absolutely. It is way more complex than I first realized it was. And it's super cool. So please learn about it and do it at your house if you can. It seems like a really cool process. So then final question for you is, what is one fun fact that listeners wouldn't know about you if all they had was your research?
So a fun fact about me is I am the in-stadium announcer and the voice for your South Georgia Tormenta FC. This is our local professional minor league soccer team, and I'm the in-stadium announcer for them. So I've always been comfortable on a microphone, and it's really fun to help, fire the fans up and get the players excited as well. So.
Oh my gosh. Yeah, you definitely seem very comfortable, which has made this a stellar interview on top of just really cool research. So I'm so glad that we had time to chat about this today, and thank you for your time and your research and trying to get that into the hands of the people who need it.
Well, thank you so much, and I'll do my traditional sign-off. It's been wonderful to be with you and to share this information with your listeners. Drive safely. Have a wonderful evening, and good night now.
Oh, man, I love that. That's so fun. That was amazing. That's probably the best outro we've ever had.
I figured you'd enjoy that.
Thank you for listening to Field Lab Earth. More information can be found in the description below. Thank you.